Yes, you're audible.
Yes, you're audible. Yeah, okay. Mhm. Is my screen visible?
Yes.
Yes. Okay. So today I will be giving a presentation on the cancer code, unraveling the role of post-translational modifications. The hallmarks of cancer comprises 6 biological capabilities acquired during multi-step development of human tumors. The hallmarks constitute an organizing principle for rationalizing the complexities of the neoplastic disease. They include sustaining proliferative signaling, evading growth suppressors, activating invasion and metastasis, enabling replicative immortality, inducing angiogenesis, and resisting cell death. Underlying these hallmarks are genome instability, which generates the genetic diversity that expedites their acquisition, and inflammation, which fosters multiple hallmark functions. Conceptual progress in the last decade has added two more emerging hallmarks of potential generality to the list. Number one is the reprogramming of energy metabolism, and number two is evading the immune destruction. In addition to cancer cells, tumors exhibit another dimension of complexity. That is, they contain a repertoire of recruited, ostensibly normal cells that contribute to the acquisition of hallmark traits by creating the tumor microenvironment. Recognition of the widespread applicability of these concepts will increasingly affect the development of new means to treat human cancer. Now we will go a little bit details into the hallmarks that I said just now, starting with the sustaining proliferative signaling. It is arguably the most fundamental trait of cancer cells and involves their ability to sustain chronic proliferation. Normal tissues carefully control the production and release of growth promoting signals that instruct entry into the progression through the cell growth and division cycle, thereby ensuring a homeostasis of cell number and thus maintenance of normal tissue architecture and function. Cancer cells, by deregulating these signals, become masters of their own destinies. The enabling signals are conveyed in large part by growth factors that bind to cell surface receptors, typically containing intracellular tyrosine kinase domains. The latter proceeds to emit signals via branched intracellular signaling pathways that regulate progression through the cell cycle as well as cell growth. That is, it increases in cell size, and often these signals influence yet other cell biological properties such as cell survival and energy metabolism. Now I will come to the second point, that is evading growth suppressors. In addition to the hallmark capability of inducing and sustaining positively acting growth stimulatory signals, cancer cells must also circumvent powerful programs that negatively regulate cell proliferation. Many of these programs depend on the actions of tumor suppressor genes. Dozens of tumor suppressors that operate in various ways to limit cell growth and proliferation have been discovered through their characteristic inactivation in one or other form of animal or human cancer. Many of these genes have been validated as a bona fide tumor suppressors through gain or loss of function experiments in mice. The two prototypical tumor suppressors encode the RB, or which we call the retinoblastoma associated, and TP53, or the tumor protein P53. They operate as central nodes within two key complementary cellular regulatory circuits that govern the decisions of cells to proliferate or alternatively activate senescent and apoptotic programs. We now move on to the third point, which says activating invasion and metastasis. In the beginning of the new millennium, the mechanisms underlying the invasion and metastasis were largely an enigma. It was clear that as carcinomas arising from epithelial tissues progressed through higher pathological grades of malignancy, reflected in local invasion and distant metastasis, the associated cancer cells typically developed alterations in their shape as well as in their attachment to other cells and to the extracellular matrix, which we also call as the ECM. And the best characterized alteration involves the loss of carcinoma cells of E-cadherin, a key cell-to-cell adhesion molecule. By forming adherence junctions with adjacent epithelial cells, E-cadherin helps to assemble epithelial cell sheets and maintain the quiescence of the cells within these sheets. We now move on to the next point, which is enabling replicative immortality. By the beginning of the new millennium, it was widely accepted that cancer cells require unlimited replicative potential in order to generate macroscopic tumors. This capability stands in marked contrast to the behavior of the cells in most normal cell lineages in the body, which are able to pass through only a limited number of successive cell growth and division cycles. This limitation has been associated with two distinct barriers to proliferation. Number one, senescence, which is a typically irreversible entrance into a non-proliferative but viable cell state. And number two is crisis, which involves cell death. Accordingly, when cells are propagated in culture, repeated cycles of cell division lead first to induction of senescence, and then for those cells that succeed in circumventing this barrier to a crisis phase in which the great majority of the cells in the populations die. On rare occasion, cells emerge from a population in crisis and exhibit unlimited replicative potential. This transition has been termed as immortalization, a trait that has been most established. That has been seen in most established cell lines in laboratories by virtue of these cell lines by virtue of their ability to proliferate in culture without the evidence of either senescence or crisis. We now move on to the next point, which is inducing angiogenesis. Like normal tissue, tumors require sustenance in the form of nutrients and oxygen, as well as ability to evacuate metabolic wastes and carbon dioxide. The tumor-associated new vasculature generated by the process of angiogenesis addresses these needs. During embryogenesis, the development of the vasculature involves the birth of new endothelial cells and their assembly into tubes, which is known as vasculogenesis, in addition to the sprouting of new vessels from existing ones, which we call as angiogenesis. Following this morphogenesis, the normal vasculature becomes largely quiescent. In the adult, as part of physiologic processes such as wound healing and female reproductive cycling, angiogenesis is turned on, but only transiently. In contrast, during tumor progression, an angiogenic switch is almost always activated and remains on, causing normally quiescent vasculature to continually sprout new vessels that help sustain expanding neoplastic growths. Now we move on to the last point, that is resisting cell death. The concept that programs cell death by apoptosis serves as a natural barrier to cancer development has been established by compelling functional studies conducted over last 2 decades. Elucidation of the signaling circuitry governing the apoptotic program has revealed how apoptosis is triggered in response to various physiological stresses that cancer cells experience during the course of tumorigenesis or as a result of anti-cancer therapy. Notable among the apoptosis inducing stresses are signaling imbalances resulting from elevated levels of oncogenic signaling, as mentioned earlier, and DNA damage associated with hyperproliferation. Yet another research has revealed how apoptosis is attenuated in those tumors that succeed in progressing to states of high grade malignancy and resistance to therapy. Post-translational modifications are an extremely important mechanism in the regulation of protein functions, and it is critical for the regulation of maintaining these hallmarks that I described in the previous slide. Now I will be briefly discussing the post-translational modifications associated with the cancer hallmarks. To begin with, I will first be discussing about post-translational modifications in regulation of sustaining proliferative signaling. Cancer cells maintain their own proliferation by dysregulating the normal physiological proliferative signaling that regulates progression through cell cycle, cell growth, cell division, as well as cell survival. Phosphorylation as a major biological regulatory mechanism is involved in almost every cellular process. Many signaling pathways I'm sorry, such as tyrosine kinase, MAP kinase, cadherin catenin complex, cyclin dependent kinase, etc. are regulated through phosphorylation dephosphorylation cascade. And the deregulation of this phosphorylation in any of pathways has been shown to be strongly associated with growth and progression of various types of cancers. One of the best examples is the epidermal growth factor receptor EGFR, a well-known protein in EGF signaling pathway and which is utilized by cancer cells to maintain the proliferative program. The epidermal growth factor receptor has served as the founding member of the large family of growth factor receptors harboring intrinsic tyrosine kinase function. High abundance of EGFR and large internal deletions are frequently observed in brain tumors, whereas point mutations and small insertions within the kinase domain are common in lung cancer. Under resting conditions, EGFR predominantly exists in an auto-inhibited monomeric form, but ligand binding confers a conformation poised to form either homodimers or heterodimers containing HER2 or other receptors. Activation of the receptor depends on the formation of an asymmetric dimer of the kinase domains in which one kinase domain allosterically activates the other. In the asymmetric dimer, the kinase domain C lobe of the activator kinase interacts with the N lobe of the receiver kinase, which catalytically stimulates the latter. Thereafter, the receiver kinase transphosphorylates specific tyrosine residues of the activator kinase. The newly phosphorylated tyrosines serve as attachment sites for various adapters such as GRB2, cytoplasmic enzymes like PLC gamma, or specific factors involved in transcription regulation. Together, these signaling factors or the effector molecules and adapter proteins link activated receptors directly or indirectly to canonical intracellular pathways as well as to endocytic machinery, which desensitizes active receptors. Importantly, several tyrosine docking sites of EGFR can bind more than one adapter or effector. Oncogenic mutant forms of EGFR mimic the ligand activated wild type form. Nevertheless, although the mutated EGFRs of tumors are enzymatically active and transforming, their tyrosine phosphorylation status is significantly lower compared to ligand activated wild type receptors. Apparently, this relatively low but persistent activity generates intracellular signals that differ from the canonical biochemical machinery. In addition, EGFR mutants frequently evade negative regulation, which we term as desensitization, such as receptor endocytosis and degradation, thus allowing the mutated receptor to act under the radar of receptor attenuating mechanism. Growth factor receptor bound 2 or GRB2 is a ubiquitously expressed adapter protein involved in several tyrosine kinase dependent signaling pathways. Stimulation of GRB2 at lysine 56 enhances the ERK activity via recruiting more SOS1 to form GRB2 SOS1 complex and consequently promoting cell migration and cell tumorigenesis. Next, we will discuss briefly about the post-translational modifications in regulation of evading growth suppression. The RB family of proteins are recruited to specific promoters through their interactions with sequence-specific DNA binding proteins. In the pathway that we will be discussing, the critical interactions are between RB pocket proteins and the E2F family of transcription factors. When recruited to E2F regulated promoters, RB pocket proteins inhibit transcription. How? By directly suppressing the transactivation of function of E2F and by recruiting factors that mediate transcriptional repression. Phosphorylation of the RB pocket proteins by cyclin D, CDK4 and CDK6 invariably disrupts the E2F RB2 binding and interaction, leading to the activation of E2F regulated gene expression. E2F binds to and regulates the promoters of multiple genes involved in cell cycle progression, nucleotide biosynthesis, DNA replication, and mitotic progression. E2F also stimulates the it also stimulates the expression of pro-apoptotic genes such as caspases and APAF1, and thus alterations in the RB pathway can affect tumor cell response to cytotoxic agents. Next, we will discuss the post-translational modifications in regulation of resisting cell death and immune escape. Ataxia telangiectasia mutated, or which we call as ATM, and ataxia telangiectasia mutant and RAD3 related kinase ATR protein kinases phosphorylate p53 to activate and enhance p53 stability. The phosphorylation of various substrates, including casein kinase 1, checkpoint kinase 1, and checkpoint kinase 2, and p53 regulate cancer cell viability by modulating many critical biochemical pathways that lead to cell cycle arrest, DNA repair, senescence, and apoptosis or cell death. P53 is mutated in about half of all human cancers, playing a critical role in tumorigenesis, tumor development, and poor prognosis. As a powerful guardian of the cell, P53 is activated by a variety of efficient and precise PTMs in response to different stress signals, regulating its stability, conformation, localization, and binding to partners to control cell cycle arrest, apoptosis, DNA repair, cellular senescence, metabolic processes, etc. P53 contains approximately 50 modification sites for phosphorylation, acetylation, methylation, ubiquitination, sumylation, ADP ribosylation, hydroxylation, and proteinylation. As the first tumor suppressor related to apoptosis, phosphorylation of P53 is a critical modification in its apoptotic cell death regulation. Ubiquitination dependent cytoplasmic localization of P53 plays an active role in apoptosis and autophagy. Acetylation also provides an important layer of P53 regulation, particularly in transcription. P53 is the first non-histone substrate regulated by acetylation and deacetylation. The acetylation of six lysine residues at C terminal, that is lysine 370, lysine 372, lysine 373, lysine 381, lysine 382, and lysine 386, enhances the sequence specific DNA binding to its target genes, including cell cycle arrest, senescence, or apoptosis after DNA damage. In DNA binding domain of P53, lysine 120 acetylation is essential for the activation of apoptosis genes such as PUMA and BAX, whereas acetylation of lysine 164 is responsible for P53 dependent cell growth arrest. Lysine 101 acetylation of P53 is critical for regulation of SLC7A11 dependent ferroptosis. More recently, it has been reported that acetylation of lysine 120 and lysine 164 is also critically involved in P53 mediated PD1 expression, suggesting a potential role of P53 acetylation in the regulation of tumor immunity. In addition, acetylation of lysine 317 negatively regulates pro-apoptotic genes and promotes cell survival after DNA damage. Besides the modification types mentioned above, a novel beta-hydroxybutyrate of P53 has been identified, which reduces P53 acetylation and P53 dependent cell growth arrest and apoptosis gene expression, such as P21 and PUMA. Different P53 mutations and their potential effects on its function and oncogenic activity is depicted in this slide. Mutations of TP53 genes is associated with inactivation of the wild-type TP53, and 75% of P53 mutations lead to loss of P53 functions. Inactivation of the wild-type function of P53 promotes invasion, proliferation, cell survival, cancer progression, and metastasis. The effects of P53 mutants are mediated by interaction with key molecular pathways that include inhibition of mTOR kinase, inhibition of CYP1, NF-kappa beta activation, stimulation of RAF MEK ERK cascade, inhibition of P53, P21, P27, and P53 BCL2 BAX pathways, inhibition of ITI-H5 tumor suppressor genes, activation of ZEB1 and ZEB2 transcription factors, and induction of epithelial mesenchymal transition. Ablation of R248Q P53 mutation in colorectal cancer inhibits STAT3 mediated tumor growth and invasion. The net effects of p53 mutations is the loss of protective effects of wild-type p53 proteins, which lead to cancer progression. Now we will briefly discuss about the post-translational modifications involved in genetic instability. To maintain genomic stability, high coordinated signaling pathways are activated upon DNA damage. DNA damage response signaling recognizes DNA breaks and arrests cell cycle progression to promote DNA repair. Alternatively, it activates pathways that lead to apoptosis or senescence in cases of extensive or irreparable damage. Upstream regulation of DNA damage response signaling is mediated by ATM, ATR, and DNA PKCs, which are phosphoinositide 3 kinase or PI3 kinase related protein kinases. ATM and ATR are the key upstream regulators of the DNA damage response signaling cascade. ATM, a key regulator of DNA damage response, is activated by double-stranded breaks and homologous recombination. The mRNA complex at the double-stranded break recruits and directly binds to and activates ATM, prompting a cascade of DNA damage response signaling via its kinase activity. The most important step is phosphorylation of serine 139 residue of the H2A histone family member X, forming gamma H2AX, which is required for the accumulation of chromatin remodeling complexes and DNA repair proteins. Subsequently, gamma H2AX recruits mediator of the DNA damage checkpoint protein and eventually triggers phosphorylation ubiquitination cascade of DNA damage response signaling. ATM also regulates cell cycle checkpoints by the phosphorylation of the checkpoint kinase 2 at G2M transition and P53 at the G1S transition. These mechanisms enable the cell cycle arrest to promote DNA damage repair or lead to senescence or apoptosis in case of irreversible damages. ATR is also an important DNA replication stress response kinase that is activated by a wide range of genotoxic stress. ATR is recruited to DNA bracket sites via direct interaction with ATR interacting protein, which recognizes RPA coated single stranded DNA that arise from the resected DSBs or double stranded breaks or during nucleotide excision repair intermediates. After localization to the bracket sites, additional factors such as DNA topoisomerase 2 binding protein 1 or Ewing's tumor associated antigen 1 ETAA1 are required to activate ATR. Activated ATR phosphorylates numerous downstream molecules with checkpoint kinase 1, which is a key target of the regulation of checkpoint signaling. Next, I will briefly discuss about a post-translational modification which is involved in maintaining genomic stability to circumvent the onset of cancers. Now, topoisomerases are enzymes which are important for releasing the torsional stress generated during any DNA transaction such as replication or transcription. Topoisomerase binds to the DNA 3' end via its tyrosine moiety and generates a nick. The DNA then swivels around the nick, thereby releasing the torsional stress, and the nick is then resealed. However, in the presence of drugs such as camptothecin and topotecan that bind at the interface between the DNA and the topoisomerase, the religation step is inhibited and thereby it generates a stable single-stranded break, which are also known as topoisomerase cleavage complexes or TOP1CC, When these top 1 CC collide with the replication or the transcription machinery, DNA double-stranded breaks are generated, which may measure by measuring gamma H2AX and which are actually lethal for the cell and must be repaired. Tyrosine DNA phosphodiesterase 1 or TDP1 is the primary enzyme involved in the repair of topo 1 associated DNA damage. It basically hydrolyzes the phosphodiester bond between the DNA and the topoisomerase, releasing the intact topoisomerase and generating a 3' phosphate. Polynucleotide kinase then comes into play and generates the 3' hydroxyl and the 5' phosphate. Polymerase beta then comes in, it fills in the gap and ligase 3 seals it. Here XRCC1 acts as a scaffolding protein. Now, as I mentioned in my earlier slides, there are a plethora of post-translational modification that govern various intrinsic molecular mechanism within the cell. And among these modifications, I will be focusing on methylation today. Now, proteins can be methylated either at their arginine or at their lysine residues. Protein arginine methylation are catalyzed by 3 types of protein arginine methyltransferases. The type 1 enzymes catalyzes the formation of asymmetrically dimethylated arginine residue. The type 2 enzymes catalyze the formation of symmetrically dimethylated arginine residue. And the type 3 enzymes catalyze the formation of monomethylated arginine residue, of which today I will be focusing on PRMT5, which is a type 2 arginine methyltransferase and catalyzes the formation of symmetrically dimethylated arginine residues. So previously, before this work was done, PRMT5 was shown to modulate the activities of FEN1, P53, and RAD9. However, how PRMT5 might be involved in the repair of topoisomerase-induced DNA damage was largely unexplored. So for exploring that, we first investigated whether there is any physical interaction between TDP1 and PRMT5, and our co-immunoprecipitation studies revealed that indeed there was a direct interaction between PRMT5 and TDP1. Next, to investigate the domains that were involved in the interaction, we generated N-terminal and C-terminal truncated constructs of both GFP TDP1 and flag PRMT5, and we did co-immunoprecipitation studies which revealed that the N-terminal domain of TDP1 interacted with the N-terminal domain of PRMT5. Then we wanted to we wanted to investigate the significance of this TDP1 PRMT5 association, and we examined whether we examined for TDP1 methylation using mass spectrometry, and our mass spectrometry studies revealed that TDP1 was dimethylated at arginine 361 and arginine 586 residues, and these residues were conserved across vertebrate species. To further validate the methylation of TDP1 on arginine 361 and arginine 586, we generated single mutants, arginine 361 and arginine 586, and also double mutant, which has both mutations at arginine 361 and 586 positions. And in methylation mutants, we observed that in case of single mutants, there was a reduction in the methylation signal on TDP1, whereas the double mutant completely abolished it. To establish whether PRMT5 is responsible for TDP1 methylation, we ectopically express GFP tagged wild type TDP1 in PRMT5 knockdown cells using small interfering RNA, and PRMT5 depletion resulted in marked reduction of the methylated band of TDP1. To further obtain evidence of TDP1 methylation, we did in vitro methylation studies using either wild type or double mutant TDP1. And we observed that in case of double mutant TDP1, the signal methylation signal on TDP1 was completely abrogated, thus suggesting that indeed the methylation of TDP1 occurs at arginine 361 and arginine 586 residues. To test mechanistic link between PRMT5 and top1 induced DNA damage, we measured poly-ADP ribose polymers and gamma H2AX in the presence of camptothecin, which is a topo1 inhibitor, as I have mentioned earlier. It actually PRMT5 deficient cells, we find that they are hypersensitive to camptothecin. And also, PRMT5 knockdown cells show marked reduction in survival rates compared to that of wild type. Consistent with the earlier data, it was also observed that PRMT5 knockdown cells had increased comet tail lengths, suggesting they accumulate increased DNA breaks. And it was also evident from confocal immunofluorescence microscopy that PRMT5 knockdown cells accumulated larger amounts of DNA breaks as estimated by gamma H2X compared to that of SI control cells in response to CPT treatment. Test whether PRMT5 has a functional impact on the 3' phosphodiesterase activity, we perform gel-based TDP1 activity assays. TDP1 catalyzes the hydrolysis of 3' tyrosyl DNA nucleopeptide substrate 14Y to a product with 3' phosphate 14P with increased electrophoretic mobility. Now, cellular extracts expressing either methylation deficient TDP1 or methylation proficient TDP1, that is TDP1 wild type, were used for the in vitro TDP1 assays, and we found that the methylation mutant TDP1 were partially defective in converting the 14Y substrate to the 14P product. Because TDP1 is found in XRCC1 repair complexes, as I have shown in my earlier slides, we tested the role of TDP1 arginine methylation on its association with XRCC1, and we find that upon double mutation or the methylation defective TDP1 did not interact with XRCC1. They failed to pull down XRCC1 as compared to that of wild type. Now, although top 1 CC reversed within minutes after washing out, when you wash out CPT, DNA damage, okay, uh, sorry, just a little bit interruption. As I shown in my last slide, it was observed that the TDP1 deficient methylation mutant TDP1 was ineffective in pulling down XRCC1, and consistent with this data, we also observed that in PRMT5 depleted cells, there was reduction in XRCC1 foci formation compared to that of cells which are proficient in PRMT5. And TDP1 methylation mutant TDP1 had less amount of survival, as we can observe from our clonogenic assay, and also they accumulated larger amount of DNA breaks as evident from our comet assay results. And then, as I was mentioning, that although topoisomerase covalent complexes, they reverse within minutes after washing out the camptothecin, DNA damage as measured with gamma H2X has much slower reversal kinetics. We therefore actually investigated the formation and disappearance of gamma H2X using immunofluorescence microscopy. As expected, levels of gamma H2AX transfected with TDP1 minus minus cells wild type, I mean, when TDP1 minus minus cells were transfected with TDP1 double mutant cells, they had higher amount of gamma H2X compared to their wild type counterpart. So basically in this experiment, what we are doing is that we are adding CPT, and as I have already mentioned in my earlier slides, upon addition of CPT, there is accumulation of gamma H2X, which you can see in all the three uh, three sets that we have taken here in case of empty vector, flag TDP wild type, or gamma H2X. However, upon the removal of CPT, we then monitor the reversal kinetics of gamma H2X, and we find that whereas wild type was highly proficient in removing the gamma H2X formation, TDP1 double mutant was defective in doing so. This suggests that TDP1 arginine methylation at arginine 361 and arginine 586 protected the cells against CPT-induced DNA damage. So taken together, this work suggests that PRMT5 interacted with TDP1 and methylated it at arginine 361 and arginine 586 residues, thereby increasing its association with XRCC1, which is a scaffolding protein, which I've mentioned earlier, and also helping it TOP1CC repair. So this work was actually published in Nucleic Acid Research. And then I come to the end of my slides. Firstly, I would like to thank my PhD supervisor, Dr. Benubroto Dash, my postdoc supervisor, Professor Margarita Santosh, and all my lab mates. And also, I would like to sincerely thank the Neotia University management, the R&D cell, the Department of Biotechnology. Now, thank you, and I'm open to questions.
Thank you, Dr. Rehman, for that very innovative and informative presentation. I am sure that our participants have gained quite a much knowledge on the cancer code and topoisomerase. So, are there any questions? So, I suppose there are no such questions.
Okay. Mhm.
Thank you once again. So, this brings us to the end of our third day session. Tomorrow, we have another lined up experts, and the details will be shared in the group as everyday schedule. So, thank you, everyone.